Yes, dear student, today we discuss another type of cell division, that is meiosis. It is also called as a reduction division. Yes, just like there is a mitosis, it has again four phases, and it is having there is a total number of a chromosome is reduced to half. That's why it is called as a meiosis. See, meiosis is a type of cell division by which germ cell, egg and sperm are produced. Meiosis is a specialized and rather complicated type of cell division which occurring only in the diploid reproductive cells and result in the formation of haploid sex cell or gametes. Daughter cells have half the number of chromosomes found in the original parent cell. Weizmann 1887 for the first time he observed meiosis. There are two types of meiosis, meiosis first and meiosis second. Meiosis first is called as a heterotypic division because of the prophase first is having a 5 substages, leptotin, zygotin, packetin, diplotin and dikinesis, and so that it is the longest duration, and so that it is called as a heterotypic division. And meiosis is just like a mitosis, and it is a homeotypic division. Similar divisions are takes place like just like that of a mitosis. So such two types we will see one by one. So prophase first, as just I have discussed, leptotin is the first subphase, zygotin is the second subphase, packetin is the third subphase, diplotin is the fourth subphase, and dikinesis is the fifth subphase of meiosis. And later on that is again metaphase first, then anaphase first, then telophase first. So these are there is a meiosis first division which takes place there is in such following subphases. So see here, this is the first leptotin phase, and this leptotin phase is there is a you are seeing in the picture is a having there is a very short stage is there, and leptotin is marked by the appearance of the chromosome as long threads. The appearance of the chromosome at this stage is a string with beads. These beads are called as a chromomeres, and it consists of condensing of the chromosome, and later on it is converted into another phase that is called as a zygotin stage. So which seen in this next diagram. So zygotin is a substage where synapses between homologous chromosome begins, and zygotin homologous chromosome pair side by side and the gene by gene with each other. This process of lateral association of homologous is called as a synapsis. So that is shown in the picture also in the diagram. When the two homologous chromosomes consisting of 4 chromatids are paired, this structure is called as a bivalent. Bivalent is there is formed in the zygotin state, and this is can be compared to a zipper like fashion. Means there is the at the end you have to see in the zip like structure is there is happening, and upper structure is having there is a open chromatids are there is a seen. So this is a looks like chromosomal appearance in zygotin. And next to there is a packetin state. Here shortening and thickening of the chromosome takes place. During this stage crossing over takes place, resulting into exchange of portion of the homologous chromosome. These has been an exchange of mother and father genetic material. As a result of variation in the genetic material, it has been increased significantly, and there is such a tetrad formation takes place. From there is a bivalent. Bivalent means there is a two bundles are there, and tetravalent is here formed, or tetrad is formed. four bundles of there is a chromosomes are there is a form. And here there is a diplostene stage. What is happening in the diplostene stage? The homologous chromosome begins to separate, particularly in the region surrounding the centromere. The sister chromatids remain attached at the centromere region. At the same point, homologous chromosomes remain in close contact. These points are known as chiasmata, which seen in the picture also. Chiasma means there is a crossing. Some chromosomes are there is a not totally separate, they are crossed with each other. The two chromosomal arms and they began to repel one another. This allows the two chromosomes to move apart from each other. They start to moving from each other. So this is a diplostene state. And the last one is a dikinesis. Last subphase of there is a meiosis first. In this last stage, characterized by shortening of the chromosomes and the terminalization of chiasmata. Terminalization means they are going to separate from each other, but somewhere they are attached. The chromosomes at their most condensed during dikinesis. The nucleolus disappears and nuclear membrane disintegrate and chromosome set free. So the chromosome, there is a nuclear membrane is going to disappear. Nucleolus is also going to disappear. The chromosome now move to periphery of the cell. After envelope is burst, cell wall is there is a nuclear membrane is there is a disappear. They are wandering and here there toward the periphery. So such type of a subphases is there is a completed, and lastly there is a metaphase. First is there is a happening. What is happening in the metaphase? The chromosomes wandering here and there toward the periphery, they came together in the center. The chromosomes themselves on the equator dissolve of the nucleus. The equatorial chromosomes are linked to the respective poles by chromosomal fibers. The continuous fibers linked to the opposite pole. The metaphase is characterized by formation of well-organized spindle apparatus. The spindle fibers are there is a well-organized and the chromosomes are toward the center line of the chromosome. And see here there is a then anaphase first. Prophase first, we have seen anaphase first is seen the picture, and this is the anaphase. What is happening in the chromosome? There is a here pair separate with sister chromatid, remaining are together, and the chromosome are pulled toward the poles due to the contraction of the chromosomal fiber. Anaphase reduces the chromosome number to half. The spindle apparatus totally disappear at the and cytokinesis starts. So this is there is a in the anaphase there is happening. And lastly telophase is happening. Telophase first, two daughter cells are formed with each daughter containing only one chromosome of the pair, and there is a cytokinin and karyokinins are there is a formed. Spindle fibers are totally absent, and in this way two daughter cells are there is a form. So see here there is some pictures of metaphase that we have just discussed, and anaphase also discussed. In metaphase the chromosome line up as we have just in the center, and in the anaphase they move toward the opposite direction. So these are the shown in the pictures. This is the telophase, cytokinin and karyokinesis there is a happening, and cell divides into two. So in this way metaphase meiosis first is there is a completed heterotypic division, and meiosis second is start, that is called as a homeotypic division. And this is a somewhat there is a similar to there is a mitosis. So what is happening here? Again there is a prophase first, there is a second is there is happening. Prophase first is there is a completed, and prophase second is happening. In the prophase second, the nuclear membrane and nucleolus are distinct in the early stages, and in the late prophase the structure disappear gradually. Chromosome are short and thick. Each chromosome is made up of two chromatids bound together by a centromere. So this is there is a happening in this, there is a prophase second. So see here, this is a picture of meiosis second. So two cells are there is a formed, and there is a metaphase second. Metaphase second, there is a nuclear membrane and nucleus both are absent, having disappeared. The sister chromatids line up along the cell equator in the center. Spindle fibers are formed. These organized into spindle. Each chromosome is made up of two chromatids held together by centromere in the metaphase. And there is a next one is there is a anaphase second. In anaphase second, the centromere break and sister chromatids separate. Nuclear membrane and nucleus are absent. Cytokinesis begins. Centromere that holds two chromatids splits. Each chromatid now has an individual centromere. Spindle fibers contract and each chromosome now pulled to the opposite pole. So this is shown in the picture also. Then there is a last stage is called as a telophase second, in which cytokinesis reaches completion, creating 4 haploid daughter cells. Nuclear membrane reappears and surrounds the group of the chromosome. Spindle fibers disappear completely. Nucleolus reappear, formation of gamete or sex cells. So in this way, there is a 4 cells are formed, which are haploid, which is seen in this picture. Product of meiosis, 4 cells are formed. So this is the again some diagrams which are how there is a gradually meiosis is there is a happening. First interphase meiosis, first 2 cells are formed, then meiosis second is there is a 4 cells are there is a formed. Then what is the difference between these mitosis and meiosis? As we have just discussed, that is a mitosis takes place in vegetative cell or somatic cell. Meiosis takes place in reproductive cells. Second one is the cell divide once here in the mitosis, and in the meiosis cell divide in the twice. And third one is the 2 daughter cells. Genetic information is identical. 4 haploid daughter cells. Genetic information is totally different. The chromosome number is maintained in the mitosis and chromosome number is reduced in the meiosis, and so that it is called as a reduction division. And in mitosis simple divisions takes place like prophase, metaphase, anaphase and telophase. But here in the meiosis complex divisions are happening because of a prophase first is having a longest duration, and it is having 5 sub stages like pleptotene, zygotene, pachetine, diplotin and dikinesis. So that it is a complex type of a division. And the last one is there is a significance of meiosis. Meiosis is a logical necessary part in the life cycle of sexually reproducing animals, since it leads to the formation of gametes. So this is a formation of a sex cell takes place from the meiosis, and due to meiosis transformation of diploid nucleus into haploid nucleus. Then meiosis lead to the variation which provide the raw material for evolution, and constant number of chromosome in a given species is maintained generation after generation due to the meiosis. And last one is there is as a result of meiosis 4 haploid cells are formed, and which are having there is a half number of a chromosome. So this is all about there is a meiosis. So today we will stop here.